Hey everyone, welcome back to my channel. This video is going to be about human papillomavirus and how there's a cure for HPV, as it's called, using light. They say here, bravo. Shout out to Zero Kelvin. He sent me this article link and they want to talk about how they've been able to cure HPV in a group of patients. There are other related articles here, such as prevention of cervical cancer gets even better. And I think that's very important. I'm glad they put that first. Starving cancer cells eat themselves. This is true. When you adjust the metabolism of cancer cells, they tend to consume each other as a stress response. LSD has staying power. Chemistry explains why. That may be interesting to you. What's the blood-brain barrier and how do you cross it? And lastly, what do racially ambivalent lizards, Amy Winehouse, canonical amino acids, and melanoma have in common? So those are some related articles you can check out. This is published February 12th, so it was a while back. But what they did was target the signaling response within the mitochondria of the cell by targeting ALA using protoporphyrin 9 within the mitochondria. And this is showing how mitochondria can biosynthetically produce porphyrin 9. This is a new word that I've never seen before, so excuse me if I'm messing up the word. There are a lot of other things going on inside of the mitochondria besides that function, but they use photodynamic therapy to do this technique. It was not accomplished with a 100-watt bulb. It was done using a technique called photodynamic therapy. Number 2, a laser and endogenous protogenic 3 amino acid called amino levulinic acid, and some very clever science. So chemistry explains the rest, more or less. Photodynamic therapy depends on the ability of a chemical to absorb light, especially laser light, and generate energy. So what they did, I'm gonna skip down past a lot of the chemistry. Some people may have interest in that, but I want to skip down to how this system really works. This comes from Pharmacology and Experimental Therapeutics. What they did in treating HPV with photodynamic therapy is the amino levulinic acid is applied to the cervix, where it is selectively taken up by the cancer cells. After about 4 hours, protoporphyrin 9 is formed inside the cells. The cancer cells are now sensitized to light, so they're photosensitized, meaning they're able to respond to light. The affected area is irradiated with a laser which penetrates the cells in the cervix. The protoporphyrin 9 inside the cancer cells absorbs energy from laser light, which causes it to form an excited state, a transient high energy form of the molecule. So you have excitation and emission. Those are the two ways light works. For instance, if you're outside in the sun, you're getting exposure to the sun through excitation, and emission would be production of vitamin D, for instance, in your skin. The excited state protoporphyrin 9 is unstable. It reacts with the molecule oxygen, forming a highly reactive, short-lived form of oxygen called singlet oxygen. Singlet oxygen is highly toxic, and that is what kills the cancer cells. So you don't get singlet oxygen without the protoporphyrin 9 and the light therapy that they're using. Healthy cells cannot take up protoporphyrin 9. Also, I have to mention that healthy cells are not as energetic as cancer cells, so they are unaffected by the laser. That is where they have a selective treatment. This is something that is very important in cancer treatment. The more selective the therapy, the fewer side effects you'll get in your patients. So the two things that are really important when it comes to therapy and antibiotics, antibodies, many other forms of therapy, is selectivity and specificity. So selectivity is something you can see almost immediately because the cells that you want to target are dead or dying. And the cells that you don't want to target, aka your healthy cells, are not affected. Specificity is where you can determine later on which type of cancer cells you have killed or which type of healthy cells are still around. If you have your cervical cells that are healthy within the lumen that are still around, that are specific for function of the cervix, but your immune cells that are healthy are negatively affected, then your treatment wasn't as specific as it should have been. So you can look up the differences between selectivity and specificity for your own research in order to understand what I'm talking about. As far as the way that the results came out here, they used 449 Mexican women in this study. All have HPV. Some had no precancerous lesions. Others had lesions or had lesions but no active infection. In a group of women who were infected by HPV but no lesions were present, the virus was eradicated in all of those women. In the group of women who were infected by HPV and also had lesions, the virus was eradicated in 64% of the women. In the group of women who had lesions but no active infection, the virus is eradicated in 57% of the women. And I think these, the second and third group, I don't think that the difference in virus eradication is significant. I don't think that's as significant due to the sample size and the length of time they did the treatment. Most importantly, there was no damage to healthy tissue observed in this study. As we know, HPV is the most commonly sexually transmitted infection. More than 22% of the U.S. population has been infected, resulting in 6,100 cancer deaths per year. A simple, non-invasive method of getting rid of the virus could represent a significant advance in public health. So they say here, nice job. I agree. I think it was good that they used photodynamic therapy. However, it's not new. As you see here, it has been used to treat a variety of cancers. There are several cancers that have been used to treat, several cancers that photodynamic therapy has been used to treat. This has come from NCI. Let's see. Let's just skip down. What types of cancers are currently treated with photodynamic therapy? The FDA has approved the photosensitizing agent called porphyrin sodium, or Photofrin, for use in PDT to treat or relieve the symptoms of esophageal cancer and non-small cell lung cancer. Porphyrin sodium is approved to relieve symptoms of esophageal cancer when the cancer obstructs the esophagus or when the cancer cannot be satisfactorily treated with laser therapy alone. So usually laser therapy is the first thing you do. If you can't get any results with that, then you move on to the photodynamic therapy. And all this does, as I said earlier, is to sensitize those cells to light. There are limitations, and this is what I wanted to get to. I always like to see the other side. Sometimes I play devil's advocate when it comes to therapy, as you know, we should look at things holistically. So the limitation is the light needed to activate most photosynthesizers cannot pass through more than about 1/3 of an inch of tissue. That's about 1 centimeter. For this reason, PDT is usually used to treat tumors on or just under the skin or on the lining of internal organs or cavities. PDT is also less effective in treating large tumors because the light cannot pass far into these tumors. And I think another thing that happens is since PDT can't pass through large tumors, you have induced inflammation, which actually feeds the tumor more. And inflammation exacerbates the tumor, activates a lot of its stress response. They turn off their death signals, and the tumor can get bigger. So they have that referenced here. PDT is a local treatment and generally cannot be used to treat cancer that has spread or metastasized. PDT does have complications and side effects. One of them is the skin and eyes become sensitive to light for approximately 6 weeks after treatment. Patients have to avoid direct sunlight and bright indoor light for at least 6 weeks. The photosensitizers tend to build up in tumors, and the activating light is focused on the tumor. You can have minimal damage to healthy tissue. However, it will cause burns, swelling, pain, and scarring in nearby healthy tissues. Other side effects include coughing, trouble swallowing, stomach pain, painful breathing, shortness of breath, but they're usually temporary. So I wanted to click on one of these references just to see what they have to say. There's one about photodynamic therapy for cancer. This was 2002, so this is not new at all. It's just new for HPV. The next one is published in 2003, Basic Principles, Applications in Oncology, and Improved Selectivity of Photodynamic Therapy. Another one is 1998, Photodynamic Therapy, from the Journal of the National Cancer Institute. And the last one I'll mention is New Directions in Photodynamic Therapy in 2002, Cellular and Molecular Biology. So these are peer-reviewed articles from PubMed. You can find them on NCBI if you copy and paste, or you can click the link in blue to go to their abstract. As you know, if there is an article that you cannot access on PubMed or NCBI, but you want to see their data, copy and paste the title of the article. So for instance, this article here, you would copy and paste that title of the article, put it into Google, click images, and you'll be able to see images from that article. You'll also be able to match the title to the abstract, and even sometimes you can access author information too. So I hope this was helpful. Once again, shout out to Zero Kelvin for sending me this information. I really appreciate it when my subs send me something that they're interested in. And that's it for me. You all let me know what you think in the comment section. I'll leave links to both these sites in the description box. Don't forget to check that out. And I'll holler at you in the next video. Bye.